everyone, my name is Yen Han. I'm a PhD student in Professor Akasaka's lab in the Department of Dermatology at Hirosaki University Graduate School of Medicine. Before coming to Japan, I studied clinical medicine in China and had clinical experience in dermatology. This experience made me interested in understanding skin disease, not only from clinical findings, but also from their underlying mechanisms. Now, I'm studying palm hunter keratoderma related to tyrosine metabolism. For this disease, good animal and cell models are still limited, so it is difficult to study the mechanism in detail. And in my project, I'm building experimental models to understand how metabolic abnormalities may affect skin keratinization. I will introduce my research project on palmoplantar epidermal differentiation disorder and palmoplantar keratoderma. The main question of my study is simple. Why does excess tyrosine mainly affect the skin of the palms and soles? To explain this question, I will first introduce the background of PPK. Then I will show how we are using cell and animal models to study it. Palmoplantar keratoderma, or PPK, is an abnormal thinning of the skin on the palms and soles. In the clinic, PPK is easy to recognize. The skin becomes thin and hard, and some patients have pain or fissures. However, even when the lesions are clearly visible, the mechanism is still not fully understood. In particular, we still don't know why these changes mainly appear in palms and soles skin. This is also difficult to study because good experimental models are limited. To study this question, I focus on TAT pad. TAT pad is caused by pathogenic variants in the TAT gene. This reduces the activity of tyrosine aminotransferase, an enzyme that normally helps metabolism tyrosine. When tyrosine is not metabolized properly, serum tyrosine levels become very high. Clinically, patients can develop lesions and PPK. The important point is that PPK can improve when serum tyrosine is lowered by diet. This suggests that high tyrosine itself may be involved in palmoplantar hyperkeratosis. So our main question is, how does high tyrosine affect palms and soles skin? To study this in a controlled system, we first used normal human epidermal keratinocytes. Calcium is used to induce differentiation, and tyrosine is the condition we want to test. After 72 hours, we compare low and high calcium conditions with or without additional tyrosine. Then we examine differentiation markers including early markers such as clathrin 10 and late markers such as involucrin. Clathrin 10 expressions is significantly elevated in the keratinocytes cultured in high tyrosine medium. At the same time, an animal model is also important. For this model, normal rats are fed a high tyrosine diet. This allows us to create a tyrosinemia type 2-like model without using genetic modified rats. Before the experiment, suitable rats were selected and their eye and paws were checked. After the high tyrosine diet, we observed change similar to keratitis and PPK. The next step is compare control lots and high tyrosine fed lots, especially in palms and soles skin. We will study keratinization and inflammation using histology and proteomics. Going forward, we will continue to improve this lot model. Our first goal is to make the model stable and reproducible. We also want to understand how seminal tyrosine levels are related to BBK. After that, we will compare skin tissue from control lot and high tyrosine fed lots in more detail. Through protein mix, we hope to find protein related to keratinization and inflammation. In the future, I hope this study will help us understand how high tyrosine causes palm-plantar hyperkeratosis in TAT pad. I also hope it will give us new clues for understanding other difficult PBK cases. What I like about this research is that it starts from questions coming directly from patients. Although tyrosinemia type 2 is a real metabolic disease, it may help us think about a bigger question in dermatology: how metabolism affects skin keratinization. For me, this is one of the interesting points of dermatology research. Many skin diseases are visible from the outside, but the mechanism behind them are not always clear. I hope this research will help me become a dermatologist who can view skin disease from both clinical and research viewpoints.